This chapter won't be that difficult and not too lengthy either. So let's see. First, we have enzymes. Enzymes are basically proteins, which are biological catalysts for the speed up a chemical reaction. And these are globular proteins, as learned in the last chapter. And these also has like active sites to which the substrate binds. So the substrate can be anything which needs to be broken down or something that needs to be where a bond needs to be formed. So enzymes are present there are two types of enzymes, intracellular and extracellular. Intracellular are basically enzymes that work inside the cell, and extracellular are enzymes which work outside the cell. Both are produced inside the cell, but extracellular are moved outside the cell through exocytosis. We will learn about exocytosis later in the chapter. So there are like two main examples given in our syllabus, one for intracellular enzyme and one for extracellular. Intracellular catalyst. Catalyst is used to break down hydrogen peroxide, which is a byproduct of many metabolic reactions. And this is like very toxic to the cells as well. So this needs to be removed. And catalyst basically converts hydrogen peroxide to water and oxygen, which is not harmful to the cells. Then we have an extracellular enzyme, which is called amylase. Amylase is basically used in digestion carried out by extracellular enzymes. This is because the macromolecules being digested are too large to enter the cell. So what basically amylase does is that it breaks down the carbohydrate through hydrolysis into simple sugar. So basically starch is converted into its monomers and then transported inside the cells. It is secreted by the salivary glands and the pancreas for digestion of starch in the mouth and small intestine, respectively. Then let's understand how enzymes work. So every enzyme has basically an active site, and this active site is specific to its own substrate. So when you say that the enzyme has an active site specific to the substrate, you can say that it has a complementary shape to the substrate instead of saying that it has similar shape, because saying that the enzyme has similar shape to the substrate would be wrong. And this basically helps in only the specific substrate binding to the enzyme. Otherwise, any other substrate can also bind, which would create a wrong reaction. As you can see here, a hydrogen peroxide molecule is shown, and this can only bind to this enzyme right here. It can't bind to this one because the active site is different as compared to the hydrogen peroxide, which is the substrate here. And they're like basically enzymes can, as I said, that enzymes can perform two types of reactions. It can perform catabolic reactions, which is basically the breaking down of a substrate to form two other products or maybe three. And then the anabolic reaction, this is the catabolic reaction example right here. So as you can see, the substrate is just one molecule, and then this binds to the enzyme on its active site, and then this will break down into its products. And then in anabolic reaction, basically a bond is formed. And an example of the anabolic reaction would be protein synthesis and photosynthesis in plants. So as you can see, two substrates bind to the enzyme and then they join together to form a single product. Now let's see how enzymes work. So basically enzymes work by lowering the energy of a reaction. So as we know that enzymes are biological catalysts, so they basically lower the activation energy. And now what is activation energy? It is the minimum amount of energy required to start a reaction. So in case of enzymes, the amount of energy needed by the substrate to become just unstable enough for a reaction to occur and the products to be formed. So it just lowers the energy so that when water, so example of breaking of a glycosidic bond in a hydrolysis reaction, the substrate is going to bind to the enzyme, and then it is going to the enzyme is going to weaken that glycosidic bond, which will, so then when water comes and binds to the glycosidic bond, it can easily break it. So the bond is weakened and the water can easily break it in the reaction. So as you can see, without the enzyme present, the activation energy was this much. And then when you put the enzyme, the activation energy was half. So it speeds up the reaction by lowering the energy required. Now there are two ways the enzymes work, the two theories that are present in our syllabus. One is lock and key hypothesis. In this, it is said that the enzymes are like rigid structures. Both the enzyme and the substrate would be your rigid structures, and only the specific enzyme which fits perfectly into its active site will bind to the enzyme. So let me show you the example. So as you can see here, the substrate will only bind to this enzyme. If it was in any other shape, it would never bind to this because it's the exact same shape over here. Whereas we look at the other hypothesis, which is the induced fit hypothesis. This is the more modern method and the one which we use these days to describe the action of enzymes. So in this, it is said that the substrate binds to the active site of the enzyme, but the active site changes its shape slightly when the substrate molecule enters the enzyme. So this basically, the changes that happen in the, the active site of the enzyme is called conformational changes. This is the term which you need to learn as well when answering your questions. The next we have investigating amylase activity. So in this, basically, we check the rate of substrate disappearance. So how the substrate disappears in different conditions. So amylase is basically used to hydrolyze starch into maltose and glucose. The amylase functions best at pH 7 and 37 degrees Celsius. So inside the mouth and the small intestine, these both places are like neutral. So amylase and starch are combined together, and then the mixture is tested for starch at regular time intervals. So maybe after each minute, the person who's investigating would just take out a sample and then put it inside an iodine solution, like as put over here. A drop would be added to each part, and then after every minute to see how much the iodine solution gives a blue-black color. So initially, the solution will give you a dark black color, and then slowly and gradually it will stop changing its color once like a certain amount of time is reached. And then the results can be plotted on a graph to better show. Then colorimetry is another method to measure enzyme activity. This is basically, I mentioned in the last chapter as well, a colorimeter is able to measure light absorbance by passing a light through it and see how much light passes. So if the light passes easily, then there is no reaction taking place or something like that. And this is basically a better and a more accurate method of measuring, but we don't usually use this. We'll just remember the name. And then dilutions is included in paper 3, so we're not looking into that. Then we have factors that affect enzyme action. So the first would be rate. First to measure the rate would be temperature. So enzymes work at specific optimum temperature, and most enzymes work best at human body temperature, which is 37 degrees Celsius. And then at lower temperatures, the enzymes have like slower, the substrates have like slower kinetic energy. So there are like fewer successful collisions, which means the substrate molecules and active site of enzyme will collide less, and then fewer enzyme substrate complexes will form due to this presence of less energy, and then the reaction would be slow. At higher temperatures, like at a certain higher temperature, the molecules will collide with a higher frequency, and the enzyme substrate complexes will form more, as the enzyme and the substrate both have higher energy, and the bonds will be broken down quickly, and the reaction will be completed much sooner. But as the temperature, since enzymes are like proteins, they can only withstand a certain amount of temperature. So after like a certain temperature increase, the enzyme will start to denature, and its the bonds which make up the active site, which includes the hydrogen bonds or the R group, hydrophobic R group interactions, these bonds will also start to break and the tertiary structure will be disrupted. So that means that the active site will also be damaged and the substrate will be prevented from binding to the enzyme. This is called denaturation. So you can say that the temperatures above 50 degrees will cause the human enzyme or the average enzyme to denature, while at for 37 or 40 degrees, the enzyme will be working at its best level. As you can see here, the activity of the enzyme increases in a very steeply till like 35 degrees, and then there's a sudden drop. After 40, the enzyme just becomes completely denatured and the activity goes to zero. Next, we have pH, and pH also enzymes will have an optimum pH at which they operate their best. So enzymes are denatured at extremes of pH. So if it's too acidic, the enzyme will denature. If it's too alkaline, the enzyme will denature. So hydrogen ionic bonds hold this tertiary structure. We all know that. And below and above the optimum pH of an enzyme, the solutions with excess H positive ions or excess OH negative ions can cause these bonds to break. So this alters the shape of the active site once again, and enzyme substrate complexes do not form. And eventually, the pH is increased or decreased so much that the enzyme denatures completely. So let's look at an example here. So pepsin is found in the stomach. We all know that, and pepsin is used to break down proteins into amino acids. So basically, why pepsin, we all know that since the stomach has like hydrochloric acid inside it, which means it has an acidic environment. So the optimum pH for a pepsin enzyme to function would be to have a very acidic pH. So if you look at this graph, it functions well at pH 2. It's not accurate, but yeah, the optimum pH for pepsin is at 2. And then urease is found inside your, I think, intestines, and probably. So this works best at pH 6 or somewhere in between. And then trypsin works best at 8.5, which is more alkaline. And if it becomes more alkaline, the OH negative ions concentration increases, then it just denatures. So to control these pH, like when measuring your enzyme catalyzed reaction, you can use buffer solutions which keep the pH stable, even when a lot of product is formed, which can change the pH of the solution. The having a buffer solution inside your experiment would make sure that the pH stays stable and that does not affect your results. Next, we have enzyme concentration. So enzyme concentration will also affect the rate of reaction. So if there's like a higher enzyme concentration, then there will be greater number of active sites available for the substrate to bind to, and a likelihood for enzyme substrate complexes to form. So as long as there is like sufficient substrate available, the graph would look linear like this as seen. And once the substrate quantity decreases, the substrate concentration becomes the limiting factor here. And even if you increase the enzyme concentration, that would have no effect on the rate of reaction, as we all know that the substrate won't be available for a reaction to take place. Then we have substrate concentration. So when measuring this, the greater the substrate concentration is going to be, the higher would be the rate of reaction. But over here, the enzyme concentration remains fixed, but the amount of substrate increase past a certain point. However, so basically, if you keep increasing the substrate concentration, the rate of reaction will increase in a steep way. But after some time, the enzyme concentration will be the limiting factor here, as there will be limited active sites available for the substrate to bind to. So increasing the substrates would have no effect on the reaction. So the graph would become a plateau like this. It would become a horizontal line. This is because all the active sites are binded, like all the active sites are occupied. So the substrate would just queue up next to the enzyme to wait for one substrate to bind and form a reaction and then leave. And then the other substrate would come and bind and then leave. So the rate of reaction just becomes constant, or you could say just plateaus off. Next, we have inhibitor concentration. So we have two types of inhibitors in our syllabus. One is competitive inhibitor and the next is non-competitive inhibitor. The difference between the two, let's look at that. So competitive inhibitor has a similar shape to that of a substrate, which indicates that the competitive inhibitor will bind to the active site of the enzyme. And then the non-competitive inhibitor just binds to an alternative site of the enzyme. When this happens, when a non-competitive inhibitor binds to an alternative site of an enzyme, you can also call this the allosteric site of the enzyme. The term isn't included in the syllabus, but if you have seen the mark schemes, the word allosteric site is included in there. So you will say that the non-competitive inhibitor binds to the allosteric or the alternate site of the enzyme, and this basically changes the shape of the active site. So this prevents the substrate from binding to the active site at all. Now, both the job of the inhibitor is basically to slow down or stop the reaction at all, to stop basically the enzyme activity, so no enzyme substrate complexes form. And even an increase in the concentration of the inhibitor either reduces the rate of reaction or has no effect on the rate, reduces the rate of reaction, and also, and in some cases, just stop it completely. If you look at this graph, the normal enzyme, the rate of reaction for the normal enzyme is just, it keeps increasing linearly and then just plateaus off. For competitive inhibitor, it also increases linearly and plateaus off almost at the same point as the normal enzyme. So this basically shows that when the substrate concentration increases, the competitive inhibitor does not, the substrate binds to the enzyme instead of the competitive inhibitor, as the substrate concentration is more than the inhibitor concentration. This is the case for competitive inhibitors. If you look at the non-competitive inhibitor line, which is the blue line right here, it increases linearly but plateaus off way less. The rate of reaction is like around half of the normal enzyme. So this shows that the non-competitive inhibitor has a much more greater effect. So once the non-competitive inhibitor binds to the enzyme, the active site shape is changed, so substrate does not bind completely. Though some non-competitive inhibition can be overcome by increasing the substrate concentration, but not, the rate of reaction cannot be revived fully. Next, we have Vmax and the Michaelis-Menten constant. So Vmax is basically the maximum rate of reaction the enzyme reaches when you plot a graph. So over here, let's look at the Vmax. If you were to say that the Vmax for a normal enzyme, should I just draw it? I'll just draw it. Let's just draw it over here. So if I were to draw a graph, it would look like this as well. So the Vmax would be this part. Vmax is your maximum activity, the rate, maximum rate reached. So this would be your Vmax. And then Michaelis-Menten constant is basically a value derived from this graph. And it's basically the half of this. It's the substrate concentration used to compare the affinity of different enzymes for their substrates. So how to calculate the Michaelis-Menten constant? So for example, this is the Vmax right here. So the half of this Vmax would be around somewhere here. This is half Vmax. Now, this graph is basically for substrate concentration against its rate of reaction. Rate of reaction. Now, now at half Emax, we're going to see how much is the substrate concentration. So the lower the substrate concentration, the higher the affinity of the enzyme. Okay. You can also it's basically inversely proportional. So the lower, so as I told you, the lower the the substrate concentration, or lower the value of Km or Michaelis-Menten constant, the higher the affinity of the enzyme. And the quicker the reaction would be reached. Okay. What else shows? It is explained well over here as well. So we can basically when we do this, we basically when we need to compare different enzyme activities, we're going to use this method. So one activity for one enzyme is plotted here. I'm going to make another line, which would have a different. Mm, mm, both reach the same point. So for this, the Vmax is shown. And now at half Vmax, you can see that the Km is landing over here for the blue line. And for the black line, the Km would end at here. So if I were to say that this was enzyme X, the blue line was enzyme X, and this was Y, then Y would have a higher affinity. Now, why do I say so? Because it reaches half Vmax before enzyme X. Because you can see that the half Emax value or the Km value for Y is lesser than the Km value for X. Now you should also be able to identify the Vmax and the Km on the graph. I described the inhibitors in detail, but I'll go through it again. So as I said, competitive inhibitors have the same shape as the substrate. So it's just going to bind on the active site of the substrate and then just block its path, and the substrates won't be able to bind and no reaction would take place. And then for non-competitive inhibitor, this is the allosteric or the alternative binding site. You can say alternative or allosteric, your choice, and then inhibitor binds over here. This just changes the active site completely as shown. So we can tell that the substrates won't be able to bind here. So again, the enzyme substrate complexes won't form. Now the non-competitive inhibitors have two examples. One is reversible and one is irreversible. So reversible inhibitors act as regulators in metabolic pathways. So metabolic reactions must also be very tightly controlled and balanced so that no single enzyme activity can run wild and continuously and uncontrollably generate more and more of a particular product. As seen here, the substrate will bind to the enzyme 1, and then the products will form until product 3 is formed, which is the end product. Now this end product, product 3, will go and act as an inhibitor for enzyme 1 once the needed required amount of product 3 is reached. So an example would be when the body needs to regulate the amount of glucose in our body. So if there is now starch will be broken down into is it starch? No, glycogen is basically broken down in our human body. And if we say that a substrate is glycogen, it will bind to the enzyme and then get broken down till it forms glucose. Now, once the glucose limit exceeds in the body, it will go and inhibit the enzyme and then just stop the glycogen from breaking down further. And the inhibition will take place until there is more glucose needed in the body. So once all the glucose is used up in the body and there is more needed, the inhibitor, which is the glucose molecule here, will just leave the allosteric side of the enzyme, and the glycogen will further break down again, and the process will start again. So this is called the continuous feedback loop. And in IGCSE biology, we learned it as negative feedback mechanism. Now, there are like two types we can measure enzyme activity. One is immobilized, and the other is free. Immobilized enzymes are basically something that uh, we make stationary and insoluble using a lignate, and the substrate is passed over this immobilized enzyme to perform the reaction. So the advantages of this immobilization is basically that the enzyme does not contaminate the product. So it is easier to package and like sell it as well. And then the immobilized enzymes can be reused as well. So we know that the enzyme production is also like very costly, and this method just makes it more easier and like cost effective as well. Then the immobilized enzymes are more thermally stable, also not really sensitive to pH changes as well. So this means that if the product is also changing the pH of the solution, it won't affect the activity of the enzyme. So the example that is used to explain this immobilized enzyme example is the example of milk. So in milk, we have lactose sugar present, and most people not, I don't know, most people are like lactose intolerant, so they can't drink milk. So for them, there is this milk in the market called lactose free milk. Now, what this lactose free milk is that basically this lactose sugar is broken down into its monomers, which are glucose, alpha glucose, and galactose sugar. So the disaccharide, which is the lactose, is broken down into its monomers. Now, how it is done is if it was free enzyme, then the enzyme would just be mixed inside the milk, which would decrease its nutritional value as well. So using immobilized enzyme means that the enzyme won't be mixed in the milk, and the nutritional value of the milk would be saved. So you get like full nutrition as well. So this just creates lactose free milk. So the milk is just poured from inside the alignate beads bound with lactase, and this milk just passes through it, and whatever is collected in this beaker right here is lactose free milk. So this has the lactose sugar broken down. This can also be used to make other lactose free dairy products as well. And yeah, that's it. This was the shortest chapter. And alignate beads and immobilization of enzymes will be also included in your paper 3 practical, in which you have to immobilize the enzyme yourself. One more thing that I would like to add is that I was doing questions as well. So it asked that why does the mesh of this immobilized enzyme made out of cellulose? So the reason would be that cellulose is insoluble, so it is used as well. So it won't like dissolve inside your solution, whichever you are putting the enzyme inside. And yeah, that's it. If you guys have any questions, you can ask. Okay, I think that's it. Thank you guys for joining. Bye bye.
Thank you for hosting.